Hello, and thank you for taking the time to review this short video to see how Multimedia Science School can help teachers save time and deliver great lessons. Multimedia Science School is a suite of 109 teaching tools that were developed by a team of science advisors, technical staff and graphic artists. A teaching tool is a collection of interactive screens with animations, models, video clips and simulations. The tools can be used for whole class teaching by projecting them onto a screen or using them with an interactive whiteboard. Also, each tool comes complete with a lesson that explains the concepts and provides summaries of key points. They can be used independently by students to aid their understanding and encourage them to take responsibility for their own learning. The software comes in two main versions, one version suitable for 11 to 16-year-olds and the other for post-16 students. In order to give you some idea of what the teaching tools look like and how they work, I will give you a quick overview of just two of them. I will not be able to include all the teaching points for the tools or fully explain the science concepts, but it's hoped that I show you enough to see how it is possible to use them in the classroom. This is the Electropotentials and Electrochemical Series teaching tool. It has four options that can be chosen from the top of the screen. When pressing play on the introduction screen, we can see the two reactions of reduction and oxidation taking place as a zinc rod is lowered into a metal salt solution containing zinc ions. This screen can be used to explain when equilibrium is reached and that there is a potential difference between the rod and the solution. At the experiment option for this tool, we can discover the standard electropotential for a selection of metals. As you can see, we have hydrogen on the left and copper on the right. When I click on this switch to complete the circuit, we can see a positive potential difference of 0.34. We can observe the standard electropotential for other metals by moving them onto the right side of the screen. If we now change the view at the top of the screen to metal and metal, we can see the electropotential and electron flow through different combinations of metal, just by placing them onto either side of the screen. The third option is convention, and when we press play, we can see the shorthand notation for an electrochemical cell that has a zinc electrode and a copper electrode. Press play again, and the notation is stretched so that each part can be easily seen, and if you move the cursor across, you can see a description of each part of the equation. If you press play for the third time, you can see labels for oxidation and reduction in the two half cells. The final option in this tool, we can select Calculation. From here we can confirm our understanding of how we calculate the potential difference between two metals. For this example, I will choose copper for my first equation, then input the standard electropotential of 0.34 into the box. For my second equation, I will select zinc and enter minus 0.76, and then press Calculate, and our answer is checked. If I press Reset, I can then go on to calculate other combinations of metals. In this tool called Extracting DNA, we have two main views, one of isolating DNA and cutting and sticking DNA. In the first phase, we have 9 buttons down the left-hand side of the screen that animates the isolating of DNA from the rest of the cell. We can see the cells being broken down mechanically and chemically. Finally, the DNA precipitates out of the filtrate using cold methylated spirit. The next option screen challenges the student to remember all the steps involved in the isolating of the DNA from a cell. After the extraction of the DNA, the tool progresses to a detailed view of a section of DNA. The way in which different restriction enzymes cut the DNA in specific locations is then explored, before moving on to look at the action of ligase enzymes in sticking fragments of DNA back together again. The student is then challenged to pair up the 3 fragments of DNA on the left side of the screen with their complementary partners on the right, using ligase enzymes. Then the student assumes the role of a genetic engineer and has to select combinations of restriction enzymes in order to extract particular fragments of DNA from a plasmid. If you would like to assess the teaching tools in Multimedia Science School, then please email Perry Lee or complete the form at the bottom of the page.